professor and vice chair of research and faculty affairs. And she's also director of the Superfund Research Program there. She received her MD with honors from the School of Medicine at the University of Granada, and then completed medical residency at the University Hospital La Paz, where she completed rotations in special topics, including epidemiological surveillance and clinical research. And during that time, she completed her MPH from the National School of Health in Madrid. Then she moved to Washington, D.C., where she worked as medical resident and consultant for the Pan American Health Organization in the Division of Disease Prevention and Control. And then she moved back to Madrid as a research fellow and resident at the National Center for Epidemiology. She received a PhD in cardiovascular epidemiology from Johns Hopkins in 2005, and she was Phi Beta Kappa. She's held a number of faculty appointments in the Departments of Epidemiology and Oncology at Johns Hopkins, and was director of the Exposure Science and Environmental Epidemiology PhD track at the Bloomberg School of Public Health. She moved to Columbia in 2016, but retains her adjunct professorship at Johns Hopkins. She has a long list of awards and honors and has been keynote speaker for many international conferences. So a couple of examples: she received the Dean's Excellence in Mentoring Award from Columbia and the Outstanding Teaching Faculty Award at Johns Hopkins. And I think you'll see that in her presentation and your interactions with her today. She also has had an NIEHS Paper of the Month. Dr. Navas-Acien has many editorial duties, including being the founding editor-in-chief of Current Environmental Health Reports. She has mentored numerous pre- and postdoctoral students, master students, and visiting scholars. Of course, when one has all these achievements, she has numerous service activities as well, including review panels, and she's currently chair of the NIH Kidney, Nutrition, Obesity, and Diabetes Study Section. She's a highly productive researcher with over 334 publications, including 21 in 2022 thus far. She's currently PI on 4 research NIH grants, plus an R25 ES grant for training undergraduates. And she's involved as a co-I on 6 additional grants. So she has numerous activities it's just a delight to welcome her to speak with us today. So thank you.
Thank you for the very nice and detailed introduction. And I hope I can see everybody. I want to, it's so nice to be in person. Can you hear me? Can you hear me well like this? And online, people can hear me well also? Very good, because you know it's such a wonderful thing to be together, and it was a great. It's great for me to be here in Louisville. And so today, what I wanted to share with you is to talk about the role of metals in disease, and I think this is a topic for which we know quite a bit, but with the new advances in analytical chemistry and the new opportunities in both experimental and observational epidemiological research, I think we are getting into a new era. We are going into much more detailed, much more sophisticated information that is already helping us to improve our strategies, not only from the prevention perspective, but also from the intervention and even at the clinical level. And so I wanted to bring some of these angles through some of the examples of some of the work that I am myself involved. And I wanted to start with this slide about what WHO says that we know about environment, thinking about environment in the very specific involuntary types of exposures going through air, water, soil, work. So from that very kind of restricted and living out the lifestyles, this is what the WHO says, that we could prevent around 30% of the burden for the major diseases, cancer, cardiovascular, pulmonary diseases, if we remove those involuntary exposures. And if you look at the chemicals in particular, WHO has this list of 10 chemicals of concern, and 4 of them are metals. And so I am going to be talking a little bit about some of these, especially arsenic, cadmium, and lead. And of course, the field of metal mixtures and chemical mixtures is very important and growing. But I want to start with the individual metals because it's already complicated enough. And only if we understand those individual complications, we are able to move forward into the mixtures, more complex analysis. And I am going to focus on these chronic low levels that affect most populations worldwide, with some populations being more exposed, particularly exposed. And as was mentioned during my introduction, I was trained as a cardiovascular epidemiologist, but I also did work in training in environmental health. And I became very interested about this idea. What if we can prevent cardiovascular disease from removing involuntary exposures? The focus of cardiovascular disease prevention has been too much on the lifestyles. And I was really interested in this field. And I finished my PhD in 2005, just when the field of environmental cardiology was kind of emerging. The first time I read that term, environmental cardiology, was in editorial commentary from Environmental Health Perspectives. And then a couple of years later, I read that paper from Aruni Bhatnagar here from Louisville. And I was starting my career as an assistant professor. And I was just like, this is what I want to do. This is the area I am going to try to make it through. And it's been very exciting and very challenging also. And we were discussing some of those challenges when you start your career earlier through some of the meetings that we had. And arsenic was, at the time when I was starting, was a clear contaminant of major concern. Really widespread in drinking water. You see here the USGS data, private well information, and you can see how there is a clear connection with community water systems from the EPA data. So arsenic is really in the water. People are drinking it, especially in the western parts of the country, also the Midwest areas, Northeast. And I was really interested in knowing more about that. And also, when you look at these places that are affected by arsenic, you see that there is a relationship with these mining areas, abandoned mine areas, parts of the country that are very geologically rich, and that disproportionately affect native communities, indigenous communities across the country. So this is true for arsenic, and it's actually also true for uranium. I want to show this map of uranium because this is a new field into which we are moving. I don't have much data in terms of health outcomes for uranium, but uranium is a very important chemical metal that is totally understudied from the toxicological perspective, from the epidemiological perspective, even from the geochemistry or other earth sciences. So there is a lot of work to do in this area. And I am particularly interested, can you see oh, no, you don't, how can I see the mouse? Sorry, because, you can see it now, very good. Oh, but somehow I need to show it here. Okay, very good, very good. You can see that there is some uranium mines near here in the northern plains. And these are some of the communities that we work with. And you can see that the community water systems are also affected in this area. They also have high uranium. So if you look at if you go and you zoom into some of these communities, no, it's, yeah, don't worry. Oh, thank you. Very good. Like that, it's easier to see. Very good. If you zoom into these communities, and this is the Ogallala Sioux tribe in South Dakota, this is a place where I work quite a bit. You can see the red dots are high arsenic and uranium. These are houses. These are people who live there. This is the water they are drinking every day. High arsenic and high uranium in red. High arsenic only in blue, high uranium only, and then the low, actually the white dots. So there is substantial variability, but you can see this arc of high arsenic and uranium going on. So we are very interested into understanding what are the health effects of those daily exposures to these metals. And we are going to be starting our Superfund Center at Columbia is transitioning from studying arsenic in Bangladesh and in other parts of the US with a more focused interest in uranium and other metals exposures in the northern plains. And hopefully it will be funded very soon. I am hopeful that it should be, the Notice of Grant Award should be coming. Maybe it came while I am here in Louisville. I didn't have time to do it. And so this is exciting, and we are going to be doing a lot of interdisciplinary work. But it's really, we came here because of all the work we've been doing, leveraging our collaboration with the Strong Heart Study. When you work with tribal communities, it takes a long time to develop those partnerships. And I've been very fortunate to be working with the Strong Heart Study since 2007. So more than 10 years now. And this is a cohort study that includes not only indigenous communities in the northern plains, but also in the southwest and in the southern plains in Oklahoma. And the communities are quite different in each area. In the Dakotas, they are very rural, very spread. In Arizona, they are near Phoenix. They've seen quite a substantial development in recent years. And in Oklahoma, they live in small towns. They are affected by a high burden of disease. For instance, here you can see the rates of coronary heart disease and atherosclerosis subclinical, much higher burden in the Strong Heart Study compared to ARIC, which is another NHLBI cohorts of white and black populations. So we have real burden of disease here. And this type of research could not take place without a strong participatory component. And we meet every year in person. In the last few years, the meetings had to transition remote, but we meet with each community regularly. And of course, we have local research teams, which are incredible and the key for the success and the possibility to do the work that we do. So this is the cohort, how it started in the late 80s. The cohort was 45 to 74 years of age for 10 years. And then they've been followed for clinical disease. But at the end of the 90s, they decided to recruit a younger cohort who were family members of the original cohort. So this brings incredible opportunities for research, both epidemiological, but also even molecular, mechanistic research is possible, genetics, because we have family structures, lots of things that can be done. I say here continuous funding is critical because if you look at the years, there are gaps. This is because NHLBI had ideas of how they wanted to fund things or not. And then we get these gaps and that's really sad. But I understand it's very challenging to do this work. But now we are starting the new phase, phase 7. Next month, participants are getting examined again, which again brings more opportunities because when we contact participants, we can add new things to collect, new questions. And that face-to-face is really essential in epidemiology. From our metal perspective, we are very fortunate because we can leverage the biomarkers. And this is our conceptual framework with connecting it with clinical disease, which is our main goal. And as I mentioned, arsenic is a widespread contaminant. And the initial hypothesis for this cohort were, first of all, can we use urinary arsenic levels as a biomarker of long-term chronic exposure? And second, which is, this was the first pilot that we did. And then can we connect this, if we have enough variability in exposure, can we connect this with disease? This is 10 years of data. These are urinary arsenic measured at the sum of the inorganic and methylated species. I'll come back to this in a few slides. And you can see some people have very low, some people have high. This is in the log scale. Arizona has higher average levels, Oklahoma higher lower levels, Dakotas all across. Arsenic is a relatively short half-life, but if you drink a glass of water with arsenic every day, it doesn't matter. Your urinary arsenic is a very stable measure of chronic long-term exposure. That was the first thing we learned, which was really important to connect with long-term clinical outcomes. So this is what we did. We looked at the association between arsenic exposure and cardiovascular disease in the Strongheart study, and this is what we found: a clear increased risk, especially at levels slightly higher than the current MCL, but even it seems that there is a throughout risk, except that for coronary heart disease specifically, there is maybe a threshold. But you can see the confidence intervals are very wide. This has been the question for arsenic going on for a long time. Is there a threshold or not? And from my understanding, talking with the toxicologists and with Chris States and others, there should be potentially a threshold for thinking of how arsenic looks like, but maybe it's very low or maybe not. So we don't know yet. We know that for sure at levels slightly below the MCL, there is increased risk, and still more research needs to be done at lower levels. These findings were consistent with another cohort in rural Colorado, and also mechanistic work. I am bringing here the work from Coran Mann, who is a good collaborator in an ApoE model, which is very interesting because it's a model that has with a lipid metabolism problem, which kind of replicates in an animal model what happens in humans with a high-fat diet. Our participants here in the Strongheart study, all of them, most of them have a high-fat diet. So it's interesting to see that there is a very kind of consistent finding in the animal model. We've been able to do mechanistic work looking at echocardiographic measures, and we can see even in young people this relationship. And again, animal model replication. I got one day an email from a postdoc saying, we've read your paper, we replicated this work in an animal model. So that was kind of a nice thing to get that message one day. And I wanted to bring something about cancer because I know some of you work in cancer. So we were able, this cohort, the Strongheart study was done for cardiovascular disease, but they also have cancer outcomes. So we were able to look at the different cancer outcomes, and especially for lung cancer, it looked very clear increased risk in our population. Arsenic measured in the urine, lung cancer developing for 20 years, more than 20 years, and we can see this very, very clear increased risk. We found no association with cancers of the breast or other cancers, and some cancer, like skin cancer, is not lethal cancer, especially non-melanoma skin cancer. So we could not look, unfortunately. You need much better cancer-specific research to be able to look at those cancers. Okay, so I want to come back to this idea that I am bringing from the beginning, that these new advances in analytical chemistry have been really essential to the work that we can do today. And technology and innovation, having high quality instruments is essential. So this is our lab, our team. It was one of the key factors of why I decided to move from Hopkins to Columbia. Joe Graziano at the time was on the verge of retiring. He was in charge of this metals lab, and he kind of contacted me. And I had been at Hopkins all my life. I thought I would retire there. And then your brain starts to dream and to think, having a lab, working with the lab together side by side it's really amazing. And it's been very exciting for me, and I am very glad I took that risk because we've been able to do lots of great work, and we are very busy and doing a lot, working with lots of epidemiological studies. It's one of the key studies we're working with now is MESA, the multi-ethnic study of atherosclerosis. This is a nationwide, 6 cities are involved across the country to look at the risk factors for cardiovascular disease. So we are measuring metals. It's a lot of work in the lab. We are very busy. We are measuring 17 elements, and we are doing the arsenic speciation. And actually, during doing this work, we've also realized we can also do selenium speciation, which is really novel. And this is now a new area of research that we've added to our team. And I really want to pause a little bit here, because when you do arsenic work in the Stronghorn study, it's actually pretty easy to do, because in the Stronghorn study population, people don't really eat seafood. Seafood is not part of the traditional diets or even the modern new diets of our communities. So we don't have to worry about what happens at the bottom part of this figure. But when you transition and want to do arsenic work in general populations, things get very complicated. Because when we eat seafood, we have a lot of seafood arsenicals, some of which contribute to the specific metabolite of inorganic arsenic too. And so we've developed some epidemiological tools to try to remove that contribution by leveraging statistically arsenobutane. And that works, but maybe not perfectly. And that's what we are learning and doing right now, this work. So it's really exciting, because you need really to understand metabolism, biochemistry, epidemiology, and toxicology, lots of things to be able to do this work well. And so this is really a great time for us right now. But if you look at this is pilot data, we have much more data now on arsenic in MESA, but this is removing the effect of the seafood arsenicals. You can see that there is a lot of variability in arsenic. These are general populations, people from 6 cities across the US. But especially the people from LA, they always have the highest levels. Always have the highest levels when you live in LA. This is because there is inorganic arsenic in water in LA. So we want to know, is this variability related to cardiovascular disease? We also want to leverage all the other metals. And for instance, this is some work actually that Caitlin McGraw, who was a trainee here, she's looking into the metal mixtures. So she put together this figure with all the metals in MESA. This is preliminary. And it seems that your metal burden seems to be related to coronary artery calcification in MESA. So this is still preliminary, but still telling us that, again, metals are important for chronic diseases in most populations. So I want to move a little bit now into a next phase of my talk. And I don't have a clock around, so I hope everything is fine. I still have a lot to share, so I don't want to be too slow. But I want to go into mechanistically, what can we do with epidemiological studies? And one of the pathways that we were very interested in looking at was through blood DNA methylation and epigenetics. This is because, again, the assays that we have today are pretty well established, they are robust, and we can incorporate them into our epidemiological cohorts. We can now go back to this DNA stored in the late 80s and early 90s, and it looks beautifully bimodal, no quality control problems. It's really amazing, remarkable. And what can you do with things that people didn't even imagine at the time of the collection? And so we started looking at arsenic and epigenomics, and obviously, not a surprise, lots of targets. Arsenic, we know, induces gene expression, and it's really related with lots of DNA methylation signatures. You can see that the top signal was annotated to a gene involved in the cysteine glutamate transport and glutathione biosynthesis. Probably not a surprise given all what's known on arsenic and oxidative stress. So lots of interesting work to follow up. And what we really wanted to check is if can DNA methylation be a pathway for the connection between arsenic and cardiovascular disease. So we did an epigenome-wide screening of blood DNA methylation and incident cardiovascular disease. And then we tried to explain mediation using some statistical tools. I'm not sure if there are people here who do statistics, so I tried to give the methods for some of them who might be interested. And so from the Illumina array, we found 72 CpGs are relevant for incident CVD in our cohort. And then looking at the 315 also that have been related to arsenic, we found that 20 of them were significant, mediating the effects in the Stronghold study. The journal, when it wasn't actually is now published, sorry, this is a typo, but they asked us, can you replicate these signatures in other cohorts? And the question is, we don't really have other cohorts that have arsenic and CBD in the same way as the Stronghold study. We have MESA. At the time, we didn't have all the metadata that we have right now. But we found that 11 of these CPGs were still associated with cardiovascular disease in these 3 cohorts. So this is quite interesting also, some level of replication. And then we started to build these protein-protein interaction networks of the different methylated CPGs, and lots of really relevant pathways show up. For instance, this with the extracellular matrix, these collagen genes, and we started to look, building on this collaboration with Korenman, we try to do this idea of triangulation, interspecies collaboration, looking at from different angles. And we were able, it was remarkable. We couldn't believe it because all these pathways were differentially methylated in the arsenic exposed cardiovascular model, the ApoE model. And this is very, is it by chance? It's still too much for me to believe that these pathways are really, we can so easily replicate them in the human and the animal model. But some of these were very interesting. For instance, this the type 1 collagen, even in an in utero model, has been shown exposed to arsenic, has shown some later cardiovascular related outcomes in the animal model. The thioredoxin interacting protein, again, related to oxidative stress. So lots of pathways that are potentially interesting. During our discussions earlier that I had, this idea of epitranscriptomics came out. And so I wanted to share that we have a new project going on that was just funded. And normally we've looked into DNA and DNA methylation, kind of upstream. But we are now very interested in going kind of downstream. What happens with RNA and RNA methylation? So leveraging the new exam that is starting in a month, we are going to use this PAX gene tube so that we can collect the RNA. RNA has been very difficult to study in human populations because the stored samples are not good to use their RNA. But now with the new exam and the new technology again, we are going to be able to do that. So this is very exciting work. We are also leveraging the multi-omics, the metabolomics, and trying to do some of that more complex work. But because there is so much that can be done, I think we need to be very strategic and think very carefully, which are the key answers that we really want to answer. So I wanted to briefly talk a little bit about genetics, because I think these affect modifiers, including nutrition, which we know is very important, high-fat diet, probably very important, other co-exposures, like we really want to look into uranium, other things, comorbidities, diabetes is very important for us, seems to be playing an interacting role, very, very relevant. But genetics is really interesting, and we are at the infancy of gene environment research. Because we really need these big cohorts that have both genetics and metals and other environmental exposures measured. But the Stronghold study is really wonderful because it's this family, the family component, leverage these large pedigrees. For a family to be in the study, each family had to have at least 5 living siblings. And they need, so the offspring, the family sets are very large. So when we've done, look at this work, we've done quite a bit with arsenic genetics. And as many of you who know about arsenic, is methylated to the arsenic 3 methyltransferase. And you can see this very strong peak in this Manhattan plot of the relation of this gene. This is a metabochip that included fine mapping for arsenic 3-MT. And you can see it's actually very difficult to identify which of any is the key SNP related to methylation, because this area is in strong linkage disequilibrium, which is also very interesting in itself. But clearly, depending on your genetics, you have more dimethylated arsenic in the urine or you have less. So this must be important for disease. And we have some evidence that this is true. For instance, looking at HOMA-IR, you can see that there is an interaction between the different alleles of the index SNP and arsenic methylation. So, and that influences how much insulin resistance you have. But this is a tiny study, and we really need to do more work in this area. I didn't talk too much about cadmium. We've done a lot of cadmium work in the SOHAR study. And I wanted to bring this as an example of how relevant, important this work is. We leveraging the family cohort, we could see a strong peak doing a linkage analysis, leveraging the family information, the fact that we know who are the siblings, the second cousins, like these very extensive pedigrees. And we found this very strong peak that when you condition on the ABCC1 transport that I was talking to Bert Watson earlier, the peak went away, disappeared completely. This is a very strong, this is the strongest epidemiologic evidence that you can get that this gene is important for regulating the levels of cadmium in the urine of people. And this is a strong, small study still. So we need to do more GWAS studies of metals. There are almost none because there are no consortiums of metal, very few, there's some, but very few. And this is, we really need to put together those, all these cohorts, all these measures to be able to do this work. The SOHAR study has been involved in some collaborations and consortium projects, especially with the DNA methylation, because that has been very easy for us to work with other cohorts. But I also hope we can do more genetic gene environment work. Before I continue, I wanted to stop a little bit because all of this work and this advancing the science in this area could not be possible without the respect and the contributions, the main role that our communities play in the science. And collective leadership, valuing traditional knowledge, and accepting the research codes that tribes have developed to protect themselves from abuse in research are really important. And data sovereignty is at the core of this, this concept. There is a lot of talk about data sharing, and there is a lot of requirements for data sharing and more that we are going to face. However, working with our communities, this is something I've learned that I didn't know when I started. The data does not belong to me as an investigator or to the institutions. The data belong ultimately to the communities and the people who have provided those data. So they should have a say. They should understand where the data are and what is being done with those data. So because of past abuses, indigenous communities have developed very clear protocols. Other communities don't have that in place. But this is, I think, it's a great lesson that indigenous communities are providing all of us and the rest of society. So this is a paper that some indigenous scientists put together in Nature, kind of reminding us that data are a responsibility, not an entitlement. And the main concern is who is profiting from this research and how can the communities also benefit from these advancements in science. So again, the idea that indigenous people must also benefit from the science is growing, and it's really important. And for me as a scientist, it's something that I want to remember every day. So how can our data be relevant at the local level, regional, and of course, nationwide and globally? But this local benefit and local ownership of the data is very important. There have been big investments to improve water quality in the Northern Plains, in the Oglala Sioux tribe in particular, so that has been very good. But still, there are lots of families who rely on private wells. And so how to help those families has been a big effort. At the beginning, when we tried to do this work, people would say, oh, nobody's going to give you the money to do this work. But at the end, we managed to get this trial funded. And now it has been completed, a little bit difficult with the COVID pandemic, but we managed to finish it. And we've worked with some of our tribal partners to develop also videos, materials, to really make it as easy as possible for people to also take ownership of their treatment systems for the water. And now the preliminary data showed some success. I don't have the data yet to show you. So preventing and reducing exposure to metals in the environment is really, it's a key and very important. But some of the metals are not easily eliminated. Even if you prevent exposure today, you still have all the accumulated exposure from the past. And this is true for metals such as cadmium and lead, which have extremely long half-lives, and we don't really know how to eliminate them from the body. So I want to finish in the last few, I don't know how much time I have, like last 10 minutes, 15 minutes? Okay, great. So I've been running a bit, but this is great because that means we have more time and also for questions. So I want to go back to lead and to finish with these metals that have this long half-life, which is not the case of arsenic. Arsenic can be eliminated very fast once you prevent the exposure. But even if you prevent exposure of lead today, or even if you remove the exposure, like in the 1968 was the peak production in the US declined, but all these past exposures are still relevant today. Interestingly, 1968 is also the year where coronary heart disease rates starting to decline dramatically. Coronary heart disease rates in the US go up until 1968 and go down after 1968. And this for me was always very intriguing. When I noticed this very early on in this, when I became very interested in this environmental cardiology area, and this concept that lead could be playing a major role in cardiovascular disease, I became particularly very interested. Tried to read a lot about lead and cardiovascular disease, and what do we know, what can we do about this? Interestingly, lead is not a clear carcinogen. That makes it very different from arsenic or cadmium, which are very clear carcinogens. Lead is especially neurotoxic. That's what we know the best. We know it's nephrotoxic also. That has been known for a long time. But the cardiotoxicity of lead was kind of forgotten for a long time. But if you go to the late 1800s, it's very beautifully described in this paper from the BMJ, how lead, Saturnine gout, which is lead poisoning in workers, describes beautiful description of atherosclerosis, including the premature aging of the arterial system, connected with the renal changes, connected with cardiac hypertrophy. This is exactly what we know about lead today. It was already known such a long time ago at high levels of exposure, and then somehow this knowledge got forgotten. I have this picture here because of Saturnine. Is that Saturn god, the god of the metals, and it's believed that Goya actually, or the dark pictures of Goya are because of lead poisoning in the paint. It became this very dark and neuropsychological problems. So, but what I want to be now in the last few minutes is to be very provocative about this idea of removing metals from the body when we cannot really easily eliminate them ourselves. And in 2013, this trial was published in JAMA. To me, that was like, wow, incredible. This EDTA, which we know binds lead very strongly, and that we know eliminates lead, showed a clear benefit for cardiovascular disease, especially among people with diabetes. And if you look at the curves, the survival curves, there is a clear separation beginning. And in this trial, what they are doing is they are giving intravenous EDTA once a week. So you go, you get a dose of intravenous EDTA for 1 week. All the participants had an MI to enter the trial. So this is a secondary prevention type of study. But a year, this is months since chelation. A year of chelation is 12 months. Look at the curves. They continue to separate. That's not common in any cardiovascular treatment. If you stop your statin, you stop your blood pressure medication, the curves don't continue to separate. They go back together. So this is quite unique. And so I was very fortunate. Tony Lamas gave me a call based on some of the lead papers I had published and asked, what do you think about this? And I thought, I think we need to study if lead is involved in this. And so we did some more pilot work and see what happens if you give EDTA to random people who have an MI. People who are like the ones in the study. What happened in the study is that this trial didn't store a single biospecimen. You know, clinical trials are very difficult to run. Multi-centers, it was a $30 million or $40 million trial, not cheap to do. But they didn't store anything. So we couldn't go back and ask what happens to lead body burden. But if you take random people and you give them EDTA and they have an MI, this is what happens. Your levels of lead go up by 5,000% because everybody has a lot of lead in their bone. Cadmium goes up by almost 500%. People, we have a lot of cadmium in our bodies because we cannot eliminate cadmium. We accumulate it. It's just too similar to zinc. It just stays in the body, in the liver, in the kidney, and any other soft tissue. And lead is just too similar to calcium, so it stayed in all our calcified tissues in the bone. So now the NIH, the FDA decided that EDTA would not be approved to treat cardiovascular disease based on a single trial. So the NIH has funded the new second trial, Dept, and now, fortunately, there is a biorepository. And I am very happy to be the PI of the biorepository. And so all the samples come to Colombia from all the places, and then we transfer them to the CDC for the meta-analysis, which is being done at the CDC. And sorry, because this picture is a little bit dramatic, and I wanted to give a little introduction to it, but I didn't have time, sorry. This is people who have critical line ischemia. This is a very serious complication of diabetes. Most people, some people without diabetes can also develop it, but most of the time it happens in people with diabetes. It's not treatable. It doesn't really get better. Most of the time it just continues to evolve into amputations. It's very damaging for the life of the people who suffer it. When we, in some pilot study, this is like, we have now like 14, I think in total cases. When they get EDTA, you can see there is a clear improvement of the vascularization. This is very remarkable. When I saw these pictures the first time, I couldn't believe it. But not only that, the people feel better. They just feel better. They can walk, they have less pain, they can sleep better, because pain during sleep is one of the big problems of critical line ischemia. So this is now something we really want to continue. This is what we see in the arteries that we see. We can see that EDTA is improving the vascularization in the arteries that we can see in these patients. So this tells us that probably something is happening in the arteries that we cannot see in the heart, in the coronaries, and the brain, and other organs. So this is really, this has been a really very interesting new area of research. I really started as a arsenic cardiovascular, metals cardiovascular epidemiology, but my career has brought me into many different angles, including being involved with these clinical trials, which is really a fascinating opportunity. So we really want to push this evidence into society. So doing that social, moving the social and clinical impact, trying to look at how can we bring the message out. It's really an effort that for me, it's important. So I invest quite a bit of time in working. For instance, I did this work with the EPA to put together a white paper for the economists at the EPA to see if we can help advance with the regulation of lead in the air and a lot of other work. And finally, I bring a slide here on e-cigarettes and metals. This is work that I had to choose, and I couldn't bring everything, but it's a work that we are very interested in doing. We've shown that e-cigarettes can release quite a bit of metals into the aerosol. And so we are now having new two new R01s looking at the effects of e-cigarettes in young adults for cardiopulmonary disease. And we've recruited more than 200 participants in the last year, which has been really exciting. So we hope to continue to do this work. So in summary, I only brought a few things. This is an area of work that is, there is a lot going on, but metal exposures are widespread, disproportionately affect some communities, and really the evidence supports their risk factors for numerous diseases. There is a lot of work that needs to be done. I think the mechanistic work provides lots of opportunities for discovery, especially for interventions. I am really eager to see more of this chelation work in animal models. I think that could be very interesting to see what's happening really, and why people with diabetes benefit more. Is zinc involved in that? Cadmium? Lots of questions. There are lots of things to do. So I hope that with the analytical technology and all the new ideas, the interdisciplinary work, we can move this science forward. And just to finish, I wanted to go back. I trained as a cardiovascular epidemiologist. Lifestyles was the basic of my training, but this idea that we can bring in the environment into cardiovascular disease prevention. What is true, I think in cancer, it has always been recognized how important the environment is. But in cardiovascular, especially in the clinical world, there is a lot of things to do. And just I want to acknowledge all the funding that supports the different studies, lots of collaborators, as you know, epidemiology, and this, not only epidemiology, these types of interdisciplinary work, it's lots of people, lots of institutions, amazing students and trainees who do incredible science. And I just leave you with this last picture of our metals group at Columbia. And I'm happy to take questions and have a discussion now. Thank you.
Very nice talk, very fascinating. Two questions. One is, we have talked about briefly about VOCs and all that, but that data is not there. But have you looked for the interaction? Sorry, have you looked at the interaction of metals with, say, like PM2.5 and see like what is the impact?
So we've done a little bit of that work. In the SOMA study, we don't have air pollution data. We are trying to bring in the models from the EPA. And the problem is that those models are particularly bad in rural areas. And so we are starting that. The reviewer said, the grant said that it's not important, that we should be studying other things than air pollution, in our computation. So I'm like, okay, thank you. We need to be more creative of how we convince that that's important to do. But in MESA, they have amazing pollution data. So we are able to do some preliminary work. It doesn't look like there's an interaction. It looks like independent effects, more or less. So So there, one hypothesis that I have myself is that part, oh, do you need to?
I should be able to hear you here.
That the one hypothesis that I have myself is a part of the effect of the particles is related to the metals in the particle. So that's something that we are interested in doing that work. Surprisingly, there is not so much work done on how much air pollution contributes to metal exposure. People know that the particles that contain more metals seem to be more toxic for the cardiovascular system. So but it's more like a mediation, more than an interaction per se. The same with tobacco smoke. We can see that the metals, cadmium especially, seems to explain quite a bit of the part of the cardiovascular effect of tobacco. But there is not, if you look at our metal exposure and tobacco, there is not really an interaction. It's more like a mediation.
And another question was like, which is really, I mean, I'm really fascinated by your EDTA, we're applying the chelation part. Have you looked at some of biomarker of revascularization, which could be used as a predictor for future events or things like that?
Yes, so the original trial, as I mentioned, didn't have any biorepository to do that. The new trial has a biorepository, so we'll be able to do that type of work. The TAC2 finished the treatment phase December of 2021, and it's going to be unblinded in the summer of 2023. So once the trial is unblinded, we'll be able to look into those types of mechanistic work through the biorepository. And so that I think is going to be really fascinating. So all of you, please, if you have ideas for ancillary studies with the TAC2 trial, because that's a resource, and if the trial is positive, you know, we don't know yet. I only think the DSMB hasn't shut us down. That's a good sign after, you know, the trial started in September 2016. So it's already a number of years. So either if it was null, I don't know, maybe they would have shut us and they would have make us wait. I don't know. But once the trial is out, if you have ideas, please contact me or Tony Lamas, who is the main PI, because lots of questions can be added to this trial. Yeah. Yes.
His life as a bedside nurse, I worked on the Navajo Nation, and I worked with a lot of diabetics with very extreme amputations. And I was wondering if you were able, because you are connected, have you been able to bring in some improvement from your connections with the strong heart so they can benefit from being intact too?
Yes, so thank you so much. In the summer of 2017 or 18, I made Tony Lamas, the PI of the study, come with me to South Dakota. And it was an amazing trip. And he appreciated it very much because, you know, and we tried really hard to have a site in the Dakotas. We needed to find some committed physicians who wanted to do it. And we tried, and unfortunately at the end, we didn't manage. So I hope that we can still do it in a way so that we can, if the study is successful, that we can do better with the implementation phase. Because yeah, it's very important.
That's an amazing talk. So I was interested in the chelation study as well. So EDTA will pull down not only those two, but also zinc, magnesium. So what do you think would be the long-term effects of pulling down the essential metals?
So the people in the study, everybody gets a mineral supplement. So they are getting magnesium and zinc as a supplement so that not to interrupt the regular stores for the essential metals. I don't know, it will be very interesting to see what happens to the essential metals through the biorepository, because we'll have blood and urine to see what are the actual changes to the metals, to the essential metals. We know that zinc metabolism is really affected in people with diabetes. We know that people with diabetes lose more zinc in the urine. Maybe the EDTA is helping with that zinc metabolism. I am not sure how, but I was fascinated. When I met with Bert Watson, I was looking at that. I was preparing my meeting with him, and I read some of the work that has been done here with a high-fat diet, an animal model with high-fat diet and cadmium. And I was fascinated because the high-fat that animal had more cadmium in the liver, and they seem to have less zinc, or there was a problem with the zinc metabolism and more cadmium. And so it made me think, maybe, what if something gets solved by removing that extra cadmium from the liver? And that allows zinc to work better in people. I don't know. So far, but it's the only thing I can, because otherwise you would think it will be harmful. But the trial didn't show harm at all. So it's an open question, but very interesting.
Thanks for bringing up liver environmental liver disease. So we've published a couple of times in these associations between lead and cadmium and apple, and we discussed one study with cadmium, just others. But if it's not already planned to do some type of liver analysis as an ancillary study in TAC2, if you're not already doing that, then
TAC2, unfortunately, adding ancillary studies to TAC2 was very difficult. We tried, for instance, to add a cognition ancillary study. And just to add a simple battery test for cognition was very difficult. So to add a liver imaging, for instance, component would have been, was not possible at the time. That we have. The biobank blood will be, so we'll be able to do some metabolic profile that hopefully can capture surrogate measure of the liver. Yeah.
Cohort studies. It looks, my IB is not up to par, but it looks like you're doing like time frames. So it's like a shot in time and you get the data and then you move on to the next. So is there any chance of following somebody who has the disease and continuing through? Because I'm thinking more of like multi-generational effects, especially when you're talking about arsenic and even cadmium, in which maybe there's the in utero exposures. And so I'm just wondering if there's any chance to that within these indigenous studies.
Yes, so what we are, let me try to go back to the, see, so the cohort, because it's multi-generational, we know who are the parents and who are the children. Yeah, sorry. So many of the parents were recruited in the main cohort, and this children are in family cohort. So what we are trying to, we've already done some work connecting, what if you look at the arsenic of the parents and you look at the risk of diabetes in the offspring, and do you know what? The association is stronger than the baseline arsenic, than their own personal arsenic. That's, I have to choose what to present, and I didn't talk about that, but that was shocking. Not only that, it seems to be related to more adiposity in the offspring. So it seems that it's more a HOMA-IR also, more insulin resistance. And that seems to be, so that's really fascinating. And again, it talks about this persistent effect. Even if you remove the exposure of today, you remove the arsenic of today, but if you were exposed in utero, then there might be some programming effect that is long lasting. So that's exactly what we are trying to answer in the new Superfund Center. So that hopefully we can do that.
And then the other question I had is kind of shifting back to the EDTA, since everybody's really interested in that. It was, Mayuk asked a really good question, actually exactly the same question I had, but on top of that, because you are giving supplements, what do you think the supplements are playing a positive effect? So I also think of, not that you can have it now, but a cohort in which you just provide the supplements to see if they also start to
So,
qualify.
because the investigators were very interested in that specific question, this is actually a 2-by-2 trial. And so one arm, there is a placebo, EDTA, placebo supplement, and high dose supplement. There is low dose supplement for everyone. But then there is a high dose supplement that is placebo supplement, and then both of them together. Last time, they didn't find an interaction, and they analyzed it just comparing the EDTA versus the non-EDTA. But even though the interaction was not significant, actually, it seems that the benefit was a little bit stronger in the people who got the supplements and the EDTA. So I don't know if this is going to replicate or not in the new trial. When the new trial was funded, there was a lot of discussion of, can we have a better, more efficient design? And at the end, the NIH and the investigate and the FDA, everybody thought, you need to replicate the trial as the first one, because if we don't find, this is a replication trial. And so hopefully we'll be able to replicate, and we'll see what happens with those supplements, if they provide any benefit or not. Yeah. Although most trials of supplements have found no effect or potential harm. Especially I remember these selenium trials and some other trials. I don't remember any zinc trials or magnesium trials, so I don't know what will happen. Thank you.
So on the lines of the supplement, with the individuals that receive supplement, do they check to see if they're already deficient in one of the minerals before giving them the supplement? Because now you're just bumping them back up to the recommended amount.
The first trial didn't check for deficiencies. In the new trial, we'll be able to do, because we have the biorepository, so we can take the baseline samples and see if people who are deficient or low do they benefit more or not? That could be awesome.
Yeah, I find that in utero exposure very interesting, because we showed 15 years ago for cardiovascular disease that in utero alone is sufficient to accelerate.
Yeah, so that's very, very important. Very important work.
Any questions online? Dr. Helmut?
All right, great.
Thank you.